Good day, good day. So today we're basically going to do DNA decode of life. So we're going to focus mainly on DNA, RNA, protein synthesis. So what is DNA? DNA, we know, is a part of that carry hereditary information. Okay, that's a sum of it. Carries hereditary information. We have different kinds of nucleic acids. We have DNA and RNA. So DNA stands for deoxyribonucleic acid, and then we have RNA, which stands for ribonucleic acid. So the two kinds of nucleic acid are found in a cell, namely DNA and RNA. These two nucleic acids are made of building blocks or monomers called nucleotides. DNA has 4 different nitrogenous bases, which consist of adenine, thymine, guanine, and cytosine, while RNA also has 4 different nitrogenous bases. There's adenine, guanine, and cytosine. So in RNA, we do not have thymine, but we have uracil, right? So now the DNA structure, it is double-stranded and it is double helix, while the RNA structure is single-stranded. So differences is that DNA is a double-stranded molecule, while RNA is a single-stranded molecule. DNA contains of deoxyribose sugar, while RNA contains of ribose sugar. DNA contains of thymine, while RNA consists of uracil. So now we're going to DNA replication. So DNA replication takes place in interphase before mitosis or meiosis begins. DNA replication, it is a process during which a DNA molecule makes an exact copy or a replica of itself. So meaning that the DNA, so this is what we're going to talk about, the DNA. So now the DNA is whereby we're going to talk about DNA replication. So DNA replication is whereby the DNA that we see here, that we see here, it's going to make a replica or an identical structure of itself. So it says the DNA double helix will unwind both strands and zip, and then weak hydrogen bonds between nitrogenous bases and 2 DNA strands and zip, meaning or they separate again. Its original DNA strand serve as a template on which its complement is built. Again, I say 3 nucleotides build a DNA strand onto each of the original 2 DNA by attaching to the complementary base pairs, meaning adenine joined to base T, which is thymine, and then guanine to cytosine, and then we have this results in 2 identical DNA molecules. Each molecule consists of 1 original strand and a new one. So meaning there are 2 replicas of DNA that are formed. So significance or the importance of DNA replication. We know that DNA replication is important because it doubles the genetic material, so it can be shared between the resulting daughter cells during the cell division, again, results in the formation of identical daughter cells during meiosis. Meaning also when it says results in formation of identical daughter cells during meiosis. So in the first meiotic division, it results in identical daughter cells that have been halved, meaning the number of the first meiotic division is halved from 46 set of chromosomes to 23. So meaning that for the correct somatic cells for the developing fetus. And then you have DNA profiling. It says every person expects identical twins has his or her own unique DNA profile, meaning every person only has their own unique DNA profile except twins, identical twins. It can be described as an agreement of black bars representing DNA fragments of the person. It is used to identify criminals, identify dead bodies, identify relatives, and identify paternity, meaning parental guy. And then we're going to protein synthesis. So protein synthesis is a process which proteins are made in each cell of an organism to form enzymes, hormones, and new structures of cells. Meaning here it is in protein synthesis we form proteins that are formed by enzymes and so forth. So what we understand that in the process of protein synthesis we have DNA translation. So DNA translation, it is the formation of mRNA and so forth. So we have DNA translation that occurs in the nucleus. So we know that the DNA starts as, remember when we said the DNA replication, it starts as the DNA replication starts, as that it says the DNA double helix unwinds, both strands unzip, weak hydrogen bonds between the strands break again, and then we say, instead of saying two like the DNA replication, we say one strand acts as a template, and then we say using free nucleotides from the cytoplasm, complementary base pairs are formed again using what the RNA molecules, meaning base adenine is joined to uracil now and cytosine to guanine. Remember now we are creating mRNA, so mRNA that is part of RNA, you see, and then it says the complementary base pairs are formed, adenine is joined to uracil, guanine is joined to cytosine. As they are now joined, now what mRNA is formed, then the mRNA leaves the nucleus using the messenger pore again, and then we now go into what translation that occurs in the ribosomes. According to the mRNA codons, tRNA brings along matching anticodons together with the required amino acids. The amino acids are joined by a peptide bond to form the required protein. So meaning again that is the process of what? Of protein synthesis. So we have codons and anticodons. So codons are formed by mRNA, while anticodons are formed by transfer RNA, meaning tRNA, right? So remember I said DNA translation says according to mRNA codons, tRNA brings along matching anticodons together with the required amino acids. The amino acids are joined by a peptide bond to form the required protein molecule. So now we have two bonds in DNA code of life. We have a weak hydrogen bond. Weak hydrogen bond joins the nitrogenous bases, while peptide bond joins amino acids. So it's two different things. Weak hydrogen bonds for nitrogenous bases, and then peptide bond for acids, the amino acids, right? And then we go to DNA mutation. So mutation that we speak of in DNA is like when now the not anticodon but the acids change. So it changes when the acid change, meaning it's going to change everything. So it says the DNA mutation goes as when the DNA nitrogenous bases change, it changes the mRNA formed, the tRNA formed, okay, and it also changes what? The acids that are formed. So a new protein molecule is what is also formed. So in that sequence, meaning when the nitrogenous bases of the DNA change, is going to change the mRNA that is formed, the tRNA that is formed, and as they change, the amino acids also are going to change. So as the amino acids are changed, we're going to have what? A new protein molecule. So the molecule that has been created when in the formation of translation, it becomes different due to what? The changes of what? Of nucleus, nitrogenous bases and so forth. So we'll continue with the second session, whereby we will be now explaining and going forth with the DNA and so forth. Thank you.